All right, so let's go ahead and talk about chapter 21. 21 is relatively a short chapter. We're going to be talking about the lymphatic system, and we're basically going to introduce lymphatic structures and kind of function of what they do. And that's a nice little intro to chapter 22. Which will be immune system. So you will kind of, here we're going to focus on kind of what is what, and then in chapter 22, we'll add some more stuff, but also we'll see how these structures are functioning. So the lymphatic system is composed of your lymphatic, hold on. Ugh. Remember how I always say I need like a second monitor or something. I have too many windows on here. But the lymphatic system is composed of lymphatic vessels, lymphatic structures, and then a fluid that fills the lymphatic vessels. That's called the lymph. So I'm going to start talking about lymph first, and then we'll talk about the vessels and kind of how the lymph flows through and what it does. So the lymph comes from the interstitial fluid. It is a fluid that is going to be, by composition, it's going to be exactly the same as the interstitial fluid. It's just the name difference comes when the fluid changes locations. So if it's in the interstitial space, the fluid is called interstitial fluid. And then as some of this fluid moves into the lymphatic capillary, it becomes lymph. So we changed the name of that. So if you remember from capillary exchange, in capillary exchange, we talked about how during reabsorption, so you have filtration into the interstitial space, and then you reabsorb most of that fluid that was filtered out. You know that some of that fluid is going to be absorbed by lymphatic system, and this is what that lymph is. So it's interstitial fluid that moves into a lymphatic capillary. Fluids is going to move into what we call a blind-ended or closed-ended lymphatic capillary. Okay, so lymphatic capillaries are blind-ended or closed-ended. And the image that you're looking at right now, this is kind of a general overview of the structures that are involved in the lymphatic system. So you have some lymph vessels that are labeled here on the left side, and then lymphatic tissues and organs are on the right side there. So we'll look at the tissues and organs more closely later. So let me share another image, and this is why we had a little bit of a delay because my file that I used to have, my PDF file, I used to have all these images nice together, and I don't know if I accidentally erased it or what happened. So let me show you the other image. And so you can see what the lymphatic capillary looks like. So I'm looking for the lymphatic capillary here. And that's this one. All right. So here you see the image of the lymphatic capillary. And notice that lymphatic capillaries are located in the capillary beds. So you've got to have this blood capillary bed in the area so that the lymphatic capillary has something to absorb, right, or to take in. So where you don't have lymphatic capillary, or where you don't have blood capillaries, you would not have lymphatic capillaries in those areas either. So lymphatic capillaries are closed-ended or blind-ended. As I said, those words mean the same thing, and it simply means that they have ends. So lymphatic capillaries are not all interconnected in the same way that the blood capillaries are. So you see that those little green things, they have ends. Okay, that's the blind end or closed end of that capillary. Let me see if I can circle that for you here. Okay, but like right here, I don't know why it picked green color for me, but right here, those are those blind ends. So they're not all connected. They have ends, almost like branches on the tree.
I have a question.
Yes.
So what happens to the lymph or like any of the stuff that is collected by the capillaries? Do they just kind of like float around there and then go back up to the nodes?
Yes. So these lymphatic, Yeah, so the lymphatic capillaries are going to eventually merge into larger and larger vessels. And then these vessels will eventually merge into what we call trunks, and then those trunks will empty into ducts. So there is going to be like a progression from the little tiny capillaries into larger and larger lymphatic vessels. And then you're going to have lymph nodes throughout the system. Does that kind of answer what you were asking?
Yeah, basically, I was just wondering like if anything special goes on those like ended parts, on like the blind
No,
end or the, no, they just kind of like, okay.
yeah, it's just one that kind of a structural difference between, because it's not a, like a circulatory system it's kind of a loop, right? And it's going to the heart. Here it's not really a loop. It's just these capillaries absorb this fluid, then larger and larger vessels ultimately will bring it back into the blood, the main bloodstream.
Okay.
Yeah.
Oh.
Yeah, we're not going to see lymphatic capillaries in avascular tissue. So again, if you don't have the blood capillaries, you're not going to see lymphatic vessels there. You are not going to see lymphatic capillaries in the red bone marrow and also in central nervous system. So those are three. So be aware of three places where you do not see lymphatic vessels. And again, those are red bone marrow, central nervous system, and avascular tissue. Okay, so that's kind of an important thing to know. Let's keep talking about the lymphatic capillaries. So we said that they're blind-ended, so structurally that's kind of a different thing compared to what you see with the blood capillaries. There are some other differences between the lymphatic capillaries and blood capillaries. And that is that the cell arrangement in the lymphatic capillaries is overlapping. So you can see on this image here, especially when you're looking at just a single one, right? The part B of this image, you see the cells are not like this next to each other, but they're overlapping, okay? And this is, imagine that there is fluid surrounding this capillary that you're looking at right now, and you add fluid into this area, you increase pressure, okay, in that area. As the pressure increases, it's going to push these overlaps open, and fluid will be able to flow into that lymphatic capillary, okay? Try to kind of imagine that as you're adding fluid into this area, it's going to push itself into the lymphatic capillary through those overlaps, through those openings. Hold on, I didn't have chat up here. Okay, just wanted to make sure that I don't have another question. And so that's kind of how the cells are arranged in the lymphatic capillary. Notice that there's not really a basement membrane in the lymphatic capillary. We know that in the blood capillaries, you have the single layer of endothelium and you have a basement membrane surrounding it. But lymphatic capillaries, you have the endothelial cells that are overlapping, and there's no basement membrane in lymphatic capillaries. So since there is not a basement membrane, we have to make sure that the cells kind of maintain that shape of the capillary. And so there are these, what we call anchoring filaments. Those are those little purple lines, and those are holding the cells, the endothelial cells of the lymphatic capillary together so that the shape of that capillary is maintained. So that's the structure of these lymphatic capillaries. So lymphatic capillaries, as I mentioned, are going to merge into larger vessels. So it's time to switch a picture. And these larger vessels will then ultimately merge into lymphatic trunks. And then those trunks will merge into a duct. So let's see, this is the image right here. So this is a pretty useful image here for you. There are several different trunks that we have, and as far as what they all drain, you should be aware of kind of like where they receive lymph from. One big thing about this image here is look at the little green man on the bottom right, okay? and his different shades of green. Okay, these different shades of green represent the area that the different ducts are draining. And when we say drain, it means that this duct is receiving lymph from that area, or it's collecting lymph from that area. So your right lymphatic duct is only going to receive lymph from your upper right quadrant. So your right side of the head and neck, right side of your thorax, and your right arm. Everything else, the rest of your body, is going to be drained by thoracic duct. So lymph from the light green area, this is what thoracic duct is going to be collecting. So it's not equal. All right. I will have some questions on here, not on here, but on the exam that will ask you, like, which of the following are drained by thoracic duct, or which of the following are drained by right lymphatic duct. So make sure that you are familiar with this figure here, that the right lymphatic duct, it's only upper right quadrant. Okay, if you remember that, and the fact that everything else is thoracic duct, then you are good. The lymphatic vessels, the larger lymphatic vessels, and even the trunks are going to resemble veins, your blood veins, in that they have those three tunics that veins had. Okay, and they also are going to have valves. Valves in veins serve to prevent backflow of blood. Valves in lymphatic vessels are going to serve to prevent backflow of lymph. You want lymph to move in one direction, ultimately towards the ducts and towards the blood vessels, so we can return this fluid back in. And on this image also, you can see the lymph nodes, some lymph nodes at least here. So on the very first one, and I forgot to point that out better on that image there, but you can see on the woman where all the vessels and structures are labeled, you can see that lymph nodes are located in different regions in clusters. And this one, as you see this vessel right here, notice that as the lymph would flow, and I don't know if you can see can you see my pointer here, the little X or plus sign? Okay, as the lymph would go this way, it would pass through a bunch of these lymph nodes. And then you have clusters like in the armpit area. So those are your axillary lymph node or various places, but the lymph nodes are always going to be kind of in clusters. And the purpose of that is that you're not only running lymph through one lymph node. The more lymph nodes you pass the lymph through, the more likely it is that if there is a pathogen, it will be detected. Okay, so you want to run it through several checkpoints. Okay, so that's why we see cluster arrangement. So there's no pump for the lymphatic system, and we know that there are valves to make sure that the lymph flows in one direction. The lymphatic vessels are going to rely on skeletal muscle pump and respiratory pump, just like your blood veins. Okay, so as your skeletal muscle is contracting, it's squeezing not only those veins, but it's also squeezing your lymphatic vessels and moving lymph through. Sometimes contractions of the smooth muscle in these vessels, because they do have smooth muscle just like veins, helps with the movement of lymph, as well as if the lymphatic vessel is near a really large artery that pulsates, sometimes that pulsing can also help move the lymph a little bit. But main mechanisms are just like veins, the respiratory pump, and the skeletal muscle pump to move the lymph through. So let's look at the trunks. So the trunks are here in the dark letters. And so some of them have left and right side, some of them don't have left and right side. So I'm gonna skip the left and right parts, just going to say jugular trunk, okay? But jugular trunk is left and right. So you would have one on left side and one on the right side. But in general, jugular trunk is going to drain head and neck. Okay, so know that jugular trunk drains head and neck, or what this means, it collects lymph from head and neck. The subclavian trunk drains breast, the superficial thoracic wall, as well as upper limbs. Then we have, as we're moving downwards, there's bronchomediastinal trunk. I skipped the duct. Okay, so I'm looking at the trunks now. The bronchomediastinal trunk is going to drain the deep thoracic structures. Then moving down lower, intestinal trunk and lumbar trunk. The intestinal trunk is going to drain structures that are within the abdominal cavity, and lumbar trunk is going to drain pelvic organs, abdominal pelvic wall, and lower limbs. So lumbar trunk is going to be the lowest, okay, or drains the lowest structures if you're thinking about like what they are draining. All right, so make a list of these trunks and be able to tell like which one drains what. Just, you don't have to add any more detail than what I just said, okay? But know that there are five types of these trunks. These trunks will ultimately merge into either the right lymphatic duct or thoracic duct. Notice thoracic duct is pretty long and it runs through your thorax, but the right lymphatic duct is very, very short. This one might be tricky to find on the visible body. So you might have to actually be hiding things and such. Yeah, you might have to play around with that one a little bit, the right lymphatic duct. Both of these ducts, or each of these ducts, is going to return lymph or return this fluid into the bloodstream, right there where the subclavian vein and the jugular veins are merging, or split, yeah, merging, if we're talking about the direction of the blood, right? So at that merger of the subclavian and internal jugular is also where the ducts are connecting on each side. And so this fluid will eventually become, part of blood again. So that's our trunks. Notice that one other structure I didn't point out here on this image is the cisterna chyli. Cisterna chyli is like a cisterna means literally like cistern or like a container. And so this is a wider area at the base of the thoracic duct. And it is specific by containing a lot of lipid-rich lymph. And this is because the intestinal trunk is bringing all the lymph from the digestive system. And the way that our body absorbs lipids is actually we're running it through the lymphatic system. And that will make a little more sense when we look at the digestive system, and you'll see how once we break down lipids, we package them into these little structures known as chylomicrons, and then they will be absorbed by the lymphatic capillaries in the digestive system. So they don't go directly into blood, they have to go through the lymphatic system. And biggest part is because the structure is too big to enter the blood capillary, but it can squeeze between the openings of the lymphatic capillary. So cisterna chyli has a lot of lipid-rich lymph. All right. So those were, so those are the vessels and capillaries and all that fun stuff. Let's go ahead and start looking at the structures, the lymphatic structures. So the first one, actually, before I start talking about specific structures specifically, first we need to know that we can divide the lymphatic tissues and organs into primary structures and secondary structures. Make sure that you know how to classify which one is primary, which one is secondary. Okay, primary structures, their function is to produce and mature lymphocytes. Okay, so formation and maturation of lymphocytes is the function of the primary structures. There are only two things that are primary lymphatic structures, and they are red bone marrow and thymus. Everything else is a secondary structure. Secondary structures don't produce lymphocytes, they don't mature lymphocytes, they simply serve for housing of these lymphocytes. So they provide a place where lymphocytes like to hang out. If you remember from chapter 18, we when we're looking at parts of blood. Hold on, I have to plug in my computer really quickly. Don't want it to die. But if you remember from chapter 18, when we looked at parts of blood, and you had the centrifuge blood, and we had the plasma, buffy coat, and erythrocytes, all the immune cells, the white blood cells, were part of the buffy coat, and that was a very small part of your whole blood. So what that means is that white blood cells, even though there are some normally in the blood, their preferred location is somewhere else. They like to be in tissues. And so secondary lymphatic structures are where you have a high concentration of lymphocytes. Secondary structures include lymph nodes, spleen, tonsils, and mold, which is mucosa-associated lymphatic tissue. So let's go ahead and start with the primary structures. The primary structures, again, are red bone marrow and thymus. The red bone marrow, you probably remember a little bit seeing something like, okay, it was not exactly like this, but we know that red bone marrow is a site of hemopoiesis. So this is where all your formed elements are formed. And so once the formed elements are formed, they will enter the bloodstream. And all of the formed elements are going to become part of blood, or they're going to travel to other places if they need to, except for your pre-T lymphocytes. And so the lymphocytes, they're formed in the red bone marrow, but they have a form or name of pre-T lymphocytes. The T lymphocytes will need to migrate to thymus. Okay, so remember, T lymphocytes for thymus. All right. And it's kind of weird how they did it on this image. It says like T lymphocytes mature in the thymus prior to circulating in blood, I think is what it says. But they are going to enter bloodstream. They're just not fully functional. Their destination is thymus. Okay, they still get to thymus through your bloodstream. Okay, the pre-T lymphocytes will then enter thymus. And then in the thymus, they will undergo selection. And so this is like a form of maturation. And we'll go over the steps of this maturation on the chapter 22 video. So it's detailed there in chapter 22, what the thymus is doing. All right. And then if they survive the thymus visit, then they can become T lymphocytes in your bloodstream. And I say if they survive because, kind of a spoiler alert, out of all the pre-T lymphocytes that enter thymus, only 2% will actually survive that selection. So it's pretty intense selection process there in the thymus. Thymus doesn't play around. It will kick out the pre-T lymphocytes that are not good for our immune system. So, and it's important to be able to sort out immune system cells that are able to do their job without damaging your own body. Okay, that's kind of the whole purpose of thymus. All right, so that's red bone marrow. Let's go to the next one, next structure that's primary, and that's thymus. All right, so thymus, here's an image of a child's thymus versus an adult's thymus, and that's a diagram. And then below that, you have a micrograph of each. And you can see that the micrographs look quite different between a child and an adult. So thymus is located in your superior mediastinum. It will be located kind of over where your aortic arch is and pulmonary trunk. So it's almost like sitting on top of the heart there. And it's responsible for maturation of T lymphocytes, right? It continues to grow. It's fairly large in children, and it continues to grow until puberty. And then during puberty, it goes backwards. So it undergoes through a process known as involution. I don't think that's in your textbook, but basically what this means is the thymus is getting smaller. So the whole organ is going to shrink. And not only does it get smaller as far as overall size, but internally, some of the thymic tissue will be lost and replaced by adipose. And so when you look at part C of this image here, all that white stuff, that's no longer thymic tissue, that's adipose tissue. Okay. Whereas in the child, you don't really see that. All of that purple and pink, that's all thymic tissue. Okay. Thymus is a bilobed organ, and then each lobe is divided into lobules, or it consists of lobules. And those are very well visible on the adult's thymus. Okay, so you just see those individual lobules there, those purple areas. You can see that each purple area has a darker edge and a lighter center. The darker edge is the cortex of the thymus, and or not cortex of the thymus, cortex of the thymic lobule. And then the center is the medulla of a thymic lobule. So thymus as a whole organ is not divided into cortex and medulla. Each lobule is divided into cortex and medulla. Yeah, makes sense? Maybe. So that's the thymus. And during the selection, like when the thymus is selecting for the T lymphocytes that are good, there's going to be part of that selection that happens in the cortex. And then if they survive the cortex, then they're going to move to the medulla. And then something else happens in the medulla. Again, that will make a little more sense when we look at chapter 22 and we talk about the selection of those T lymphocytes. Okay, I guess that's about it. Yeah, I'm not talking about the selection here. So that's about it for the thymus. Let's look at the secondary lymphatic structures. Probably the most intense would be the lymph node, because there's a bunch of things that you need to be able to label on it, and then spleen, and then the rest is not really that big of a deal. Okay, so let's look at the lymph node here. Here it is. So this is a typical, yeah, what did somebody say?
I was like, okay.
Okay. So this is a typical lymph node, or an image or diagram of a lymph node. Some things that are important to point out. Look at how many vessels are bringing lymph to the lymph node. So if you look at the green arrows, that signifies the direction of lymph. And these vessels that bring lymph to the lymph node are known as afferent lymphatic vessels. And I really try to emphasize the A versus the E part, okay? So afferent is the entry, or they bring the lymph to the lymph node. Then each lymph node has a hilum. Hilum is this concavity, this concave area of the lymph node. And out of this concavity, out of this hilum, you would see generally just one efferent, E for exit, lymphatic vessel. Sometimes there might be two, but usually there's just one, okay, that takes lymph out of the lymph node. And this allows the flow of lymph to, be slow enough for the lymph node to filter it and look for any pathogens and such. Lymph node is surrounded by a capsule. The size of lymph nodes can be anywhere from 2 millimeters to about an inch in size. Inch is about 2.5 centimeters. So they can be very, very tiny, but they can be, fairly large as well. The lymph node is surrounded by a capsule. Under the capsule, you would have subcapsular space or sinus. Is that labeled? That's not labeled on here, but you might see, if you're looking at either diagram somewhere else, you might see subcapsular sinus. And then the lymph will flow between these kind of a purple and pink nodules that you see in the cortex. So internally, lymph node is divided into cortex and medulla. Cortex is that purple pink area with those nodules. Okay, that's the cortex. And then the center, the yellow and purple that you see there, that's the medulla. Okay. Under the microscope, generally you would see that cortex is more dense and medulla is less dense, like there's more white space in the medulla. So this is, you have a picture there in the textbook image. There's also a picture of a lymph node in the histology file that you can look at, and you'll see that the nodules are only going to be around the outside of the lymph node, but you don't see them in the medulla. And you can kind of see that on this micrograph here as well, that the nodules are only around the outside, but not in the center. So the lymph will flow between spaces between the lobules, and those spaces are called cortical sinuses. And then from cortical sinus, the lymph will flow through the medulla through medullary sinus. If we look closer at the cortex, okay, and you can see that the top part B of this image right there, the top that says cortex, that's looking at one lymphatic nodule, and it includes the sinus around it, okay? So you have the cortical sinus that surrounds each lymphatic nodule. Then there's a mantle zone, that's the darker part of the lymphatic nodule. And you can see that on the microscope slide there, okay? Each nodule has a darker area, and that's that mantle zone. The mantle zone is going to have T cells, and it has macrophages and dendritic cells. These cells, especially macrophages and dendritic cells, these cells are good for trapping pathogens that could be present in that fluid. Okay, so dendritic cells literally have dendrites, and they can kind of grab things and engulf things. Both macrophages and dendritic cells, the way that they act, and more on that will be in chapter 22, but just kind of like as an intro, macrophages and dendritic cells will trap a pathogen and engulf it. So like they'll literally eat it, right? And then when it's inside of that cell, they will break it down. And then when they break down the pathogen, they will show it to T cells. So T cells are important there. They're hanging out around macrophages and dendritic cells, because macrophages and dendritic cells can show a T cell the pathogen. T cells are very important, but they are not capable of recognizing a pathogen on their own. So anyway, so that's what you would have in the mantle zone, these cells that are waiting to grab onto something that's not supposed to be there. And then you have in the center, you have the germinal center. Germinal center is going to be full of B lymphocytes. B lymphocytes are cells that once they're activated, they will become plasma cells, and these plasma cells will produce antibodies. So if the T cell in the mantle zone gets activated by being shown a pathogen, it will send a signal to the B lymphocytes, and B lymphocytes will be like, okay, this is what we're doing. Let's go ahead and start producing antibodies. So that's cortex. I'm sorry, my kid is crying. I hope you don't hear that. And then, so that's cortex, and that's the lymphatic nodule. Okay, let's look at the medulla. Medulla is a little different. You don't see these nodules there. So when the lymph enters the medulla, it's going to flow through medullary sinuses, which are the spaces. And then what you see there are medullary cords. So medullary cords are like connective tissue with immune cells. So it's almost like a mantle zone. You would have these macrophages and dendritic cells. You would also see some B cells and T cells in there. So I kind of imagine it as like a structure, and a bunch of these cells are attached to it. And so as the fluid flows through, these cells then can grab onto any pathogens as they're flowing through. So kind of the same principle of the mantle zone. It's just these are arranged as cords in the medulla rather than creating a border of the lymphatic nodule like the mantle zone is doing. So that's the medulla, and that's the lymph node. So make sure that you're able to label this. I don't think it's super challenging, the lymph node. So hopefully it's all good. All right, let's talk about spleen now. And here's the spleen.
So just to kind of summarize what a lymph node does, it basically like filters lymphatic fluid with the help of those T cells, sorry, T lymphocytes and B lymphocytes, right?
Yes, yes,
Okay.
yes. So lymph node filters lymph. And now we're gonna look at spleen, and spleen also filters something, but spleen filters blood. Okay,
Mm.
make sure that you know the difference between the two. So lymph node filters lymph with the help of your immune system cells, and spleen filters blood also with help of the immune system cells. But yeah, so spleen is located in your the upper kind of like a left quadrant of your body. It's if you see where the pancreas is right here, it's kind of touching the pancreas, the tail of the pancreas there, and it's kind of turned to the side on your side. That's kind of how it would go. The spleen does have a hilum, so that's that concavity where the arteries, the splenic artery and splenic vein enter and exit the spleen. There's no cortex and medulla for spleen. Spleen does have a pretty thick capsule, and then internally it's divided into red pulp and white pulp. White pulp are the nodules, so they look similar to what you saw in the lymph node, right? The darker edge and the lighter center, but they're not the same. So here these round or oval structures are known as the white pulp. Everything else that surrounds those nodule-like looking things, the white pulp, is the red pulp. Okay, so this is, you know, the red pulp is the red stuff. Okay, and then the white pulp are those nodule-like looking things. The white pulp, so since spleen filters blood, it's important to talk about how the blood flows through the spleen. Spleen is going to receive blood from the splenic artery. Splenic artery then splits into smaller and smaller arteries until there will be a branch of this artery that is going to be dedicated to one area of the white pulp. And this artery is going to be called central artery. It's very, very small. It almost could be called arterial. And you said you do see my pointer? Right here. Yeah. Do you see it? Or no?
I went away.
Okay. So I'm going
There it
into
is.
I'm going into the picture. I'm going to show you the central artery right here. Okay, see there's a little tiny circle there, and that's the central artery of this white pulp. Okay, so right here is the central artery.
Oh my God, that's
Yeah, it's tiny. So on the
Yeah.
And we would, you know, in the lab we would actually zoom a little closer, but generally you can see a little small blood vessel somewhere nearby each white pulp. You don't see anything like this in the lymph node. The central artery will bring blood through the white pulp. It will branch into smaller arterioles. And as the blood flows through the white pulp, there are some immune system cells that are able to detect and engulf pathogens. So you would see macrophages here, B cells as well, and such. So they're there waiting to engulf anything that's not supposed to be there. As the blood flows through the white pulp, it will eventually enter sinusoids. And if you remember from chapter 20, I think 20, when we were talking about the different types of capillaries and how you know, sinusoids are kind of like a hybrid between a space and a vessel. Well, look at the sinusoids here. Okay, these are they don't look like a vessel, they look like spaces. Okay, so the shape of the sinusoid here is very different compared to what we saw in that table there. Okay, but the blood will enter the sinusoids here. You're going to have some more immune cells that are there ready to engulf pathogens, but also ready to recycle erythrocytes. So sinusoids are places where erythrocytes, worn out erythrocytes and worn out platelets can exit the bloodstream and then get recycled by your body. So you have these cells, these cells that engulf things, ready there to recycle things, break down things. So sinusoids, and then from sinusoids, the blood is collected by splenic venules. The venules will merge together into larger and larger veins, and ultimately you will have a single vein that is going to take blood out of the spleen. Okay, so where it becomes a little tricky is just in that white bulb area. Okay, central artery brings blood into the white bulb, blood flows through the white bulb. There are some immune cells that will engulf pathogens. Then eventually blood will enter sinusoids. This is where we can recycle erythrocytes and platelets. From sinusoids, the blood is collected by venules and then larger and larger veins and ultimately splenic vein and out of the organ. So that's our spleen. Almost done. This is pretty much the last picture that I'm going to show you here, and that is the tonsils. Okay, so right here you see the tonsils. I need you to be able to tell me which tonsil is which. Okay, so make sure that you know how to label this image. I will definitely ask you to label the 3 different tonsils that are important to this. So you have the pharyngeal tonsil. That's right at the top superior part of the pharynx. Kind of like on the border between the nasal cavity and the pharynx, right? So that's your pharyngeal tonsil. Then we have palatine tonsils, and pharyngeal and palatine are paired tonsils. So you have 2 pharyngeal, you have 2 palatine tonsils, and then for lingual tonsil, you only have 1. And the lingual tonsil is behind, like on the posterior side of the tongue. Okay, so lingual has to do with tongue, and palatine is right here. If you go to a mirror and open your mouth and stick out your tongue and look inside of your oral cavity, if you have palatine tonsils, you should be able to actually see them. So those are palatine tonsils. The tonsils are some people actually consider them part of mold. Because they don't have a full capsule, so it's not like a specific organ. Because the secondary structures, or really lymphatic structures, we can also divide by presence or absence of capsule. And if the structure does not have a capsule, it's considered like lymphatic tissue. If it does have a capsule, it's considered a lymphatic organ. So like spleen would be a lymphatic organ. Lymph node would be a lymphatic organ. But tonsils do not have a complete capsule. It doesn't go all the way around this, so we would consider the lymphatic tissue. What you can see inside of the tonsil, if you look at the microscope slide here, you can see that there is a bunch of lymphatic nodules. These lymphatic nodules are pretty much the same as what you would see in the lymph node. Okay, so they have that kind of the mantle zone, that outer part, the darker part, and then inside you have that germinal center, the lighter part. So structurally and as far as like what kind of cells there are, they're very similar. Location is different, of course. The tonsils protect you from pathogens that you may inhale or ingest. So look at their location. That kind of makes sense, right? So things that we could inhale or ingest, the tonsils would detect. Okay. And then, if you look at the part C there, you can see those lymphatic nodules even closer. There you have that germinal center. And then the darker part would be the same as that mantle zone in the lymph node. So that's tonsils. And then the last thing is mold. I don't have a picture for mold, but mold is mucosa-associated lymphatic tissue. And this is found in several places in your body, usually something that's open to the exterior. And that's why sometimes some people consider the tonsils as part of mold. Because this is in a cavity that's open, right? So you will see not might see, you will see these this lymphatic tissue, the mucosa associated lymphatic tissue in areas like in lamina propria of the mucosa of the, or mucosa. It's either lamina propria or mucosa. Sometimes it kind of goes through both. But your gastrointestinal or GI tract, your respiratory tract, your genital urinary tracts as well. Okay, so places that it make it easy for pathogens to enter your body. Okay, you have this mucosa associated lymphatic tissue. In your small intestine, there are these regions that are very heavy on lymphatic tissue, and they look like nodules, like the darker outside, germinal center in the inside, but they're very prominent in the ileum, especially of the small intestine. And these patches, or this lymphatic tissue, is known as Peyer's patches. So we'll see them in the digestive system. The reason why you would want heavy presence of lymphatic tissue in your small intestine, this is the main place where you're absorbing most of nutrients, most of things from your digestive system. And so you have to have some protection from possibly pathogenic things that your body would be absorbing from the digestive system. So that's our molt. Yeah, that's it for chapter 21.